Hi everyone, welcome to lesson 2.3, Biomolecules. As we go through this lesson, make sure to fill in your notes and keep up with your vocabulary terms. So carbon is often called the building block of life. Carbon atoms have 4 valence electrons, which allows the formation of covalent bonds with many other elements. Some of these elements include hydrogen, oxygen, phosphorus, sulfur, and nitrogen to form the molecules of life. Carbon can also form bonds with itself, and as seen below, carbon has the ability to form millions of different large and complex structures, as it can form single, double, or even triple covalent bonds. So remember, if you recall from last lesson, carbon is a non-metal and it was found in group 14 of the periodic table, meaning that it has 4 valence electrons and therefore it wants to form 4 bonds. Since it is a non-metal, carbon will pretty much always form a covalent bond. Carbon is unique because of this property and because it's very abundant in our atmosphere compared to other elements. All of the 6 elements from last class, the CHNOPS, are vital to life and clearly very important, but carbon gets the special title of the building block of life. The organic molecules found in living things are known as biomolecules. Many are also known as macromolecules because of their great size. Biomolecules are made from thousands or even hundreds of thousands of smaller molecules. Most biomolecules are formed by a process called polymerization, in which large compounds are built by joining smaller ones together. Now we have these two terms, monomers and polymers. So the monomer is the small chemical unit that makes up a polymer. So here in this diagram, you see monomers are kind of like the puzzle pieces or building blocks. The polymers are molecules composed of many monomers. So polymers here are kind of like the completed puzzle or the building itself. But the formation of monomers going into polymers again is known as polymerization. Monomers join together to form polymers, and the monomers in a polymer may be identical or different just depending on the biomolecule. So the 4 groups of biomolecules that we're going to discuss today are carbohydrates, lipids, proteins, and nucleic acids. So we're going to look at each of these individually in a little bit more detail. So carbohydrates are biomolecules made up of carbon, hydrogen, and oxygen atoms, and they are a type of nutrient that is the major source of energy for the body. A few functions of carbohydrates include that living things use them for energy. So the breakdown of sugars, such as glucose, supplies immediate energy for cell activities. For plants and some animals and other organisms also use carbohydrates for structural purposes. So think to yourself, what's the typical ratio of carbon to hydrogen to oxygen in carbohydrates? So the answer is clearly right there, but if you remember back to Unit 1, the formula for glucose was C6H12O6. So the ratio for carbohydrates is usually 1 carbon to 2 hydrogen to 1 oxygen. The monomer of carbohydrates are monosaccharides, and the polymer of carbohydrates are polysaccharides. So if you look down here at this diagram, the monomer or the building block is a single sugar, okay? So 1 monosaccharide, while the polymer, polysaccharides, are chains of these little sugars connected together. This one in particular, we have a glucose molecule showing as the monomer, and then the chain of glucose molecules connected together is a starch. So now let's look at types of carbohydrates, and we have monosaccharides, disaccharides, and polysaccharides. Monosaccharides are just the single sugar molecules, and they are also called simple sugars. A couple examples of monosaccharides are glucose and fructose. Disaccharides contain 2 sugar molecules combined together, and a couple examples of those are sucrose and lactose. Polysaccharides are many monosaccharides joined together, and a few examples of polysaccharides are starches, glycogen, and cellulose. So if we look here at this diagram down here, the monomer of carbohydrates is monosaccharides, okay? So monosaccharide here is just 1 single sugar unit. The polymer of carbohydrates are polysaccharides, okay? And again, that's just many monosaccharides joined together. So that's all that this picture here is trying to show is 1 glucose molecule is the building block. Many glucose molecules joined together forms that chain or the polymer, which in this case is a polysaccharide for carbohydrates. Next up, we have lipids. Lipids are biomolecules made mostly from carbon and hydrogen atoms, and they include fats, oils, and waxes. Now, lipids do usually contain some oxygen, but if you recall from that last slide, carbohydrates had that nice ratio of 1 carbon to 2 hydrogen to 1 oxygen. Lipids are typically many C's and H's and only a couple to a few oxygens. Lipids are a large and varied group of biomolecules that are generally not soluble in water. So think back and think, what does that term mean? Not soluble in water. What would we consider those? So if you're thinking to yourself, either hydrophobic or nonpolar, you are correct. So a few functions: lipids store energy, and usually they contain more energy than carbohydrates, but that energy is released more slowly. They form important parts of biological membranes. Many steroids synthesized by the body are lipids, and some steroids are hormones, which function as chemical messengers. Lipids can also form the waterproof coverings in plants, and if you recall, that waterproof covering from Unit 1 was called the cuticle. Now for the monomer and polymer of lipids, this is a little bit different. So lipids are a little different and they don't have a true monomer and polymer. Okay, so instead, what we call them for lipids are macromolecules and subunits. The reason for this is just because in order for a macromolecule to be classified as a polymer, its monomers need to be the same type. Since lipids are made up of a glycerol and fatty acid and not necessarily the same type, we can't classify lipids as true polymers. Instead, we call glycerol and fatty acids the subunits instead of monomers. So just a simple note here, again, monomer and polymer instead are just called subunits and macromolecules for lipids. So now a few types of lipids. Many lipids are formed when a glycerol molecule combines with a compound called fatty acid. So here in this diagram to the right, this is a glycerol, or what we call glycerol head, and these right here are 3 fatty acid tails. 1 piece, 2 piece, 3 piece. Okay, so 3 fatty acid tails. A saturated fatty acid is one that contains carbons only joined by single bonds. So we get that term saturated because the fatty acids contain the maximum number of hydrogen atoms. So whenever you see saturated, I want you to think that is a large number of hydrogens. Now the reason is because each carbon can make 4 bonds, okay, or it can completely fill itself with all of those hydrogen bonds. A few examples of saturated fatty acids are fats and waxes, and saturated fatty acids tend to be solid at room temp. Unsaturated fatty acids contain at least 1 double bond between carbon atoms. They contain fewer hydrogens because the carbon-carbon double bond fills an extra spot that hydrogen could have taken. So remember, carbon can form 4 bonds, okay? So again, if it has a double bond, a carbon-carbon double bond, that's taking away a bond that could have been a hydrogen to carbon bond. A couple examples are cooking oil such as olive oil, and unsaturated fatty acids tend to be liquid at room temperature. A polyunsaturated fatty acid just contains more than 1 double bond between carbon atoms, but it still kind of falls under this big umbrella of unsaturated fatty acids. So we are at our first check for understanding. Pause here, read the questions, and try these out on your own. So let's go through and check ourselves. So looking at the table, which of the 4 fatty acids is saturated and which are unsaturated? So stearic is saturated and the other 3 are unsaturated. Number 2, how does melting point change as the number of carbon-carbon double bonds increase? So melting point decreases as the number of double bonds increases. And we're going to look at the reason why for number 2 on the next slide. Number 3, if room temp is 25 Celsius, which fatty acids are solid and which are liquid at room temp? So stearic acid is solid at room temp and the other 3 are liquids. So this is continued from number 2 on that last slide. So how does melting point change as the number of carbon-carbon double bonds increase? So it said melting point decreases as the number of bonds increases. So the reason for that is the more double bonds you have, the more bends in the molecule. So if you look here at this diagram, saturated is typically all of those single bonds. Okay, unsaturated, if you have a double bond, it causes what we kind of call a kink, okay, meaning that it's going to cause a bend. As such, whenever you have many unsaturated fatty acids here with these bends, they don't stack very well. Okay, so here's an example of this. Okay, nicely stacked, solid, not nicely stacked, therefore liquid. Okay, so since it doesn't stack very well, that means less intramolecular interactions. Therefore, again, it's going to appear liquid. Next up, we have proteins. Proteins are biomolecules that contain carbon, hydrogen, oxygen, and nitrogen, and they're needed by the body for growth and repair. A couple functions is proteins control the rate of reactions or form enzymes. So we're going to talk about enzymes more so later in the unit, but remember, enzymes are proteins and enzymes control the rate of reactions. Number 2, they can regulate cellular processes, transport substances, help fight diseases, and number 3, they can also form cellular structures. An amino acid is the monomer of proteins. They have what we call an amino group and a carboxyl group on the other end. And we're going to look at the structure of an amino acid momentarily. Covalent bonds called peptide bonds link amino acids together to form a polypeptide. So the monomer proteins are amino acids and the polymer proteins are what we call polypeptides. Now from that last slide, it mentioned what we had for amino acids, recall, amino group and a carboxyl group. So the amino and carboxyl are what we call functional groups. A functional group is a specific group of atoms within a molecule that is responsible for a characteristic of that molecule. Now you don't need to memorize the functional groups, okay? So all of this information on this slide right here, please do not worry about memorizing. We just wanted you to see the table of common functional groups found in biology and kind of how you identify them. So continuing with proteins, proteins are made up of amino acids as we saw in that last slide. So here's the general structure of amino acids, and you do need to know this, okay? So for amino acids, they have the amino group, and that's this green group here, that NH2. Amino acids also have a carboxyl group, the COOH, okay, that is the carboxyl group. Also, for the amino acid, they have what we call an R group. Okay, and the part of each amino acid that is different is this R group. R groups can be acidic, basic, polar, nonpolar, and some can even contain large ring structures. So this is why proteins are considered one of the most diverse biomolecules, is because the R group is going to vary depending on the amino acid that you're looking at. Proteins have many different functions, again including controlling the rate of reactions, forming bones, muscles, or transporting substances into and out of cells. Now, we're going to briefly cover this slide. Okay, we're going to talk about levels of protein folding. For now, we don't want you to get too caught up in the details. The most important information on this slide is the protein shape is extremely important for the protein function. So amino acids are assembled into polypeptide chains according to the instructions coded in the DNA. Or what that means is the DNA is telling the proteins how they need to form. The proteins are described as having 4 levels of structure: primary, secondary, tertiary, or quaternary. The primary structure is just the chain or sequence of amino acids. The secondary structure is dictated by the hydrogen bonding of the peptide backbone, and that's going to cause a couple different patterns, either an alpha helix or a beta pleated sheet. The tertiary protein structure is determined by the 3D interactions due to the side chains, or those R groups from the last slide. And then the quaternary protein structure consists of more than one amino acid chain, or what we call subunits. Now, all proteins have primary, secondary, and tertiary, but not all proteins have a quaternary structure. So again, if that was confusing, just remember the shape of a protein is maintained by a variety of bonds and forces, some of those including ionic, covalent, and hydrogen. But the protein shape is extremely important for protein function. Last, we have nucleic acids. Nucleic acids are biomolecules containing hydrogen, oxygen, nitrogen, carbon, and phosphorus. So for functions, they store and transmit hereditary or genetic information. And a couple examples of nucleic acids include DNA and RNA. The monomer of nucleic acids is the nucleotide, and the polymer is nucleic acids. Nucleotides are the monomer, and they have 3 parts, as you can see here in this diagram. They have a 5-carbon sugar, either deoxyribose or ribose, depending on if it's DNA or RNA, a phosphate group, and a nitrogenous base. And we're going to look more so at the structure of this later on in the unit whenever we discuss DNA in more detail. So here's our exit ticket. Pause here and try to answer the questions on your own. Now let's go ahead and check ourselves. So the maximum number of bonds that carbon can form is 4 bonds. Again, that is because carbon has 4 valence electrons. When a fatty acid has no double bonds, it is saturated, meaning that it has the maximum number of hydrogens. Polymerization was just the process of monomers joining together to form polymers. All amino acids have the amino group and carboxyl group. And then that other part of the amino acid, that R group, is the part that varies. For number 5, the 3 parts of a nucleotide are a 5-carbon sugar, either ribose or deoxyribose, depending on the nucleic acid that you're looking at, a phosphate group, and a nitrogenous base. And number 6, what are the functions of nucleic acids? And that was to store and transmit hereditary information. So that is the end of lesson 2.3. Okay, go back, double check your notes, make sure you have all of your vocabulary terms down, and thank you very much for watching.